This is where we finished up earlier on today with Burkitt and bowel cancer. Now, this paper caused quite a stir in the UK, and our Department of Health noticed it and thought, has this guy really got anything? And they came to the unit I was working in, the MRC gastroenterology unit, and they said, can you investigate it and find out if there's any truth in it? I was the most junior research person in this unit in the early 70s, and the rest of them were working on high-flown gut physiology and neurophysiology and things, and the boss of the unit walked around people and said, the MRC want us to look at fibre and what it does in the gut. And universally, everybody said, go away, it's disgusting. I mean, we really don't want to get involved in this. And he finally came to me as the last person in the unit and said, well, could you look at it for me? And that's how I got into fibre. And up to that, I was doing serious science, but since then, it's been a bit iffy. Right, I'm going to move through some of this because we don't need it. Right. So the first thing, the good thing about it was the MRC were willing to give us as much money as we wanted. So we built a metabolic suite, hired the staff, and the first question we asked was, does fibre really affect bowel habit, which was one of the things Burkitt said. And we very quickly showed that it was enormously important for bowel habit or stool weight. And this slide represents the accumulation of a number of individual studies, which these days would be subject to systematic review. But each one of these is the mean and standard error for a human-controlled dietary study looking at different amounts of fibre on bowel habit. And I think whichever way you look at it, you'll think it's probably significant. The more fibre you... No, do we not? We don't have this. Okay, there's nothing to point with. On the bottom line, there is this stuff called non-starch polysaccharide, which is the purist's view of fibre. And the more you have in your diet, the bigger your stool weight. And that is an irrevocable truth about dietary fibre. And there's nothing else in the diet does it. We looked at fat, we looked at protein, and everything else. All useless. So there was something going on there. Right. Oh, yes, there's been some discussion about what is dietary fibre, and we don't really need to go into that because it's an endless argument. I saw Michael put up the Codex definition. Is he still here or has he run away? Yes. Put up the Codex definition, which Jim and I were responsible for bringing together. We agreed to the definition because Codex had discussed it for so many years, we thought, for goodness sake, let's move on from this, and you have your definition, and it's an utterly hopeless definition. Everything's wrong about it. You should never define a dietary component in terms of what it does. You should define it in terms of what it is. Fundamentally important. And the dietary fibre definition of Codex is a physiological definition. It's what escapes digestion in the gut, in the small bowel. And that is hopeless. If you try and measure it, you'll realize how hopeless it is. You must define dietary components chemically. That's a simple summary of a lot of prejudices. So non-starch polysaccharides are the main dietary fibre components. Right. So we found that fibre affects bowel habit. The question we then started asking was why and how does it work? And at the time we did this work, the suggestion was that fibre took up water, retained it in the gut, passed through the gut, and that was how it affected bowel habit. So we developed an in vitro model of water holding versus the fecal weights we had from our human volunteer studies. And these are all, each point represents the relationship between the water holding capacity measured in vitro and the change in stool weight. And we got exactly the opposite of what we expected. The more water a fibre held, the less effect it had on bowel habit. When in research things turn out to be the opposite of what you want, that's progress. So we then decided we'd look at at feces and see what was there. And to our, not astonishment, but to our surprise at the time, and these are controlled dietary studies in people on a typical UK diet, which was one of the diets, and 3 other diets with added cabbage, carrot, fibre, or bran. And you will see that fibre intake is in blue and excretion is in yellow. 80% of the fibre was disappearing during passage through the gut, and that really applies to all fibres, apart from bran, which is a bit more resistant. So what in heaven's name was going on in the gut to increase stool output and bowel habit when it was mostly disappearing? That's the proportion of increase in stool weight in relation to the different studies. So we got this electron microscopes to look at stools under the microscope. They didn't really want to do it, but they did. And here you will see that under the electron microscope, stools are just packed full of bacteria, which was what Michael's been talking about, and that's absolutely right. And there are huge varieties, cocci and rods and all sorts of stuff. The only one extra thing on this slide I bring your attention to is that a lot of them are sitting on particles. And that's very important for the efficiency of microbial metabolism, because when they're on a surface, the bugs make consortia and pass nutrients one to the other and become much more efficient. So we looked at it and we discovered that there were all these bugs there. So we developed a method for measuring the bugs in stool. And we borrowed a method from ruminant physiology. The ruminant's very interested in microbial conversion of feed, because that's part of the efficiency of the way feed benefits ruminant animals. And we borrowed a method from the ruminants, a gravimetric method, for isolating the bugs from stools and measuring them that way. And when we did this, the thing on the right is the pellet of microbiota from the stool, and the left-hand side is what actually is the composition of human feces, and the major component is bacteria. And we've done this in dozens and hundreds of studies. There is some small residual fibre, a little purple band, and then there is free sort of gel captured water. Those are the main components. And the bacteria we called biomass, because that's what the ruminant physiologists call it. That's the amount of bugs in a system. It's biomass. And that's a fly, isn't it? Yeah. So the biomass is the amount of bacteria in a system. And we went on to measure biomass in sudden death victims. And I managed to get about 40 colons from people that had died of heart attacks and various other things. And we measured biomass in the colon, and it's 43% of the weight of the colonic contents. And I've never managed to get it published. And today, I think this is what is the microbiome. It's probably very close to what is the microbiome. Only what we're talking about is a crude gravimetric method measurement, but that's what it is. We had a lot of backup from microbiology with this. Right, back to what's going on in our subjects. So here, we've got the effect of the dietary fibre on fecal biomass excretion. And on the greeny columns are the control diets, and when you add in the fibre from these different sources, up goes biome excretion, fecal biomass excretion. And now we're beginning to move towards a mechanism for how fibre affects bowel habit. And there's a 25% increase in biomass in these studies on average. The numbers here are dry matter. To convert it to wet weight, you multiply by 5, because bugs are approximately 80% fibre, 75 or 80%, something in that order. So what's happening with fibre is you're increasing biomass. Dry matter, okay. Biomass is 47% of wet stool weight if we multiply it up. So approximately half of our stools and our gut, large bowel contents are biomass. Right, so this is what's happening. Fibre goes in. We've got microbial metabolism, and it's very important. It's a unique metabolism in the body because it occurs without the benefit of oxygen. And you get biomass growth, and that goes out into feces. And one of the important things about biomass growth is it takes up nitrogenous compounds. We haven't got time to talk about that today, but that's a very important story in relation to bowel cancer. Right, we then looked at the soluble component of gut contents and built an ionogram. And on the right-hand side are the cations you find in the large bowel, principally potassium, and these other ones, a little bit of sodium and some calcium and magnesium in solution. But more interestingly, on the anion side, dominated by short-chain fatty acids, which people are now beginning to talk about today quite freely, which is good, because they are of fundamental importance to understanding how the large bowel works and how fiber works. And the short-chain fatty acids are these three fatty acids, acetic, propionic, and butyric. You can tell your mother-in-law that acetic acid's in vinegar, and she'll understand straight away what you're talking about. So you have these three short-chain fatty acids are the principal anions in the large bowel. And to cut a long story short, they come from the anaerobic metabolism of carbohydrate, and in the absence of oxygen, this is what bugs produce. And they are absorbed into the blood. 98% are absorbed into the blood. So what comes out in feces doesn't tell you very much about what's going on. And so we have what's a process in the large bowel that is fundamentally important to the large bowel and to understanding the work of fiber. And that is the fiber is broken down by the anaerobes. It produces on the right the biomass, which takes up nitrogen. You have the short-chain fatty acids in the middle, and there are also some gases that everybody knows about, hydrogen, methane, and carbon dioxide. And this is called fermentation. But unlike the fermentation in wine making, which I gather goes on in New Zealand from place to place, which uses yeast and is aerobic, this is anaerobic and uses bacteria, and the products are acids. If you produce these acids in wine fermentation, that's a bad thing, I gather. So this is a fundamentally important process going on in the hindgut. Right. Now, it's 1982, and we are joined in Cambridge, this is my group in Cambridge, by this young man at the back on the left, Ru Pamare. One of the best, he's over on sabbatical from Wellington, and he was one of the best guys we ever had in the lab. He was really, really very good indeed. And I'll show you some of the work he did. The only other thing is there's a guy, he brought over with him the guy at the right-hand side, and I don't know who that is. Now, if you work out how old he's likely to be, and if you know who it is, let me know. I totally lost. He didn't stay for the whole year or so. He only stayed for about 2 or 3 months on a student placement. But I don't even remember his name. Anyway, Ru did some very interesting studies on short-chain fatty acids. The first thing we did, we tried to measure them in blood and found that with quite a complex technique of freeze fracturing and things, we could do it. And so we took healthy volunteers, and we used pectin, which Stuart will be highly familiar with, as an example of fiber. And we studied people for over 24 hours. Both Ru and I were volunteers in this. It was a terrible study to do because you had to wake up every 2 hours in the night and take blood. So we had a control diet, which was fiber-free, and we measured the short-chain fatty acids in blood, and realistically, there's only acetate there. And over the 24-hour period, you'll see at the top, the black line is the control line, and then with the pectin, you get this increase in acetate in blood, and the bottom line is the breath hydrogen, which confirms the fermentation thing. But we actually have always had all this diabetes talk today. I don't think anybody's shown a glucose tolerance test, which I was hoping they would. Because if you had a glucose tolerance test here, it's all over in 2 hours, 3 hours. I mean, this is something completely different going on in your hindgut. But it's digestion. So you put a glucose tolerance test at the top there, by 2 hours and certainly by 4 hours, it's all finished. But it's hardly starting in your hindgut. Right, okay, there isn't time to go into this. But we then started to try and understand why there was only acetate in peripheral blood. And we took, I joined a surgical team in South Africa and took portal blood and arterial blood and these sort of things, and we got some autopsy material from recently people that had died recently and measured the short-chain fatty acids in all these different sites. So you don't need to be too careful about this now. I keep trying to point this thing. So UK autopsy, South African surgery, propionate, acetate, and butyrate, and these are the important numbers. These are the molar ratios. So you take the total amount of short-chain fatty acid and you express each one as a proportion, the molar ratio. And in the human colon, the top line there, 57% is acetate, 22% propionate, and 21% butyrate. That's what's produced in fermentation. That's an in vitro model which shows the same thing. There's a theoretical model which shows something similar. In portal blood, the notable thing is the butyrate's almost disappeared. And in arterial blood, the propionate's gone down as well. And what is happening in the human gut is the arterial blood goes to the large bowel. The short-chain fatty acids are produced by fermentation. The butyrate is taken up by the colonic epithelium, and that's very important for the epithelial health of the colonic epithelium. The propionate and butyrate get into the portal vein, but the liver takes up the propionate. And if you were a ruminant, your life would depend on that, because it's a nice 3-carbon fragment you can make glucose from. And the ruminant doesn't absorb any glucose from its gut, because it's all chewed up by the bugs. But we don't do that. So the propionate's taken up by the liver, and the acetate goes out into the peripheral blood. And there are organs that really like to have acetate, like the brain and the kidney. And if you starve people, the acetate in blood doesn't disappear. There's always a basal level of it always coming, and I presume it comes from the liver. So this is a sort of overall physiology of short-chain fatty acids in the human. Right. So I mean, this is more for the undergraduates, really, because everybody else will know this, but you've got two completely different digestive structures, processes going on in the gut. This is the traditional one, carbohydrate in the diet, pancreatic enzymes, break it down to maltose, dextrins, brush border enzymes turn it into glucose, it's absorbed, insulin secreted, goes to the liver, metabolized to CO2 and water. That's the bog standard way we all exist. But there's another system going on in the large bowel where carbohydrate which reaches this, which is so-called fiber, but it's a variety of carbohydrates, broken down by the bacterial enzymes in an anaerobic system. Short-chain fatty acids are absorbed, and they go directly to muscle and liver. There's no insulin secretion. So it's a totally and importantly different system. Right, benefits of carbohydrate, let's forget about that. Right, because I don't think I can go back, can I? I wouldn't know how to. Can I go back? Oh, okay, just very briefly. The benefits of carbohydrate fermentation in the large bowel provides hosts with energy through short-chain fatty acids, okay. Bypasses insulin secretion. Butyrate, again, this is a big story. Been around for a long time now, but it has important effects on the expression of genes, histone deacetylase, and these things, all of which point to it being protective against neoplastic change. So it's very important in the colon cancer story. And we showed this in the 80s, and it's still everybody that produces these wonderful complex diagrams of genes and things and has butyrate on it, it's still in there, and it's important. Stimulation of biomass, laxative benefits, okay. Strengthens resistance to pathogen invasion, very important. That's a whole talk on its own. But if your biomass is healthy, it has a natural resistance to invading pathogens. Nitrogen uptake is a very important story in relation to bowel cancer. It all relates to meat and the products of protein fermentation, which go on in the colon in certain circumstances. Metabolism, bioactive compounds, B vitamin system synthesis. That's it, okay. There's another dimension to this story. Right. Okay. So we've had this slide already. This is the biomass increase with the fiber in these different studies. And there's 3.6 grams, this is dry matter, additional biomass in these studies, in these patients. These healthy volunteers. Probably students, I'm sure they were. Right, okay. Have that in your head, 3.6. And we also know in the same studies how much fiber is disappearing from this. 80%, and we know the numbers. so we can work out how much biomass you get from how much fiber. Okay, here it is. Biomass yields. Fiber digested, biomass increase, and the yield in percentage terms are those along the bottom line, and the average is 29%. And if you look at the ruminant data, it's exactly the same. We are the same as all other fermenting species, apart from the termite. But this is a universal biological phenomenon, that you get a yield of about 29, 30, 33% biomass from carbohydrate fermentation. Okay? But there is another dimension to this, which again we learned from the ruminant studies, is that it's not simply bacteria and fiber, and you break them down and you get the biomass. An important thing the ruminant people taught us was that there is a moderating factor, and that's time. And it's called turnover, or in terms of the work we do, transit. And this is from, somebody was talking to me about, yes, you, Saliers, here she is. So you know this work. Shout up if you think I'm talking complete rubbish. This is work, in vitro work from Isaacson and Salier's work in the late 70s, early 80s. And they're looking at biomass yield in vitro fermentation systems in relation to turnover. And the times on the bottom line here are from 10 to 70 hours. And the biomass yields are approximately around what we are finding, but they depend very much on the rapid turnover time, the yield is higher, and the slow turnover time, the yield is lower. And that all relates, again, we don't have time to talk about it, to the needs of bacteria for maintenance of metabolism when there's not much energy substrate around. Okay, so we decided to look at transit time in relation to biomass development in the large bowel. And we took a bunch of subjects, and each one of these dots is an individual, and we put them on everybody on the same standard diet, and we watched them on the standard diet, control diet, the open circles, I think. No, I don't know which ones they are. And then we slowed down transit with a drug, codeine, and we speeded it up with a laxative, Senokot. And you will see that the biomass yield in our human studies is very closely related to transit or turnover time. People with slow transit will have much less efficient conversion of their fiber to biomass. And we can add to this little table I put up before of biomass yields, and on the bottom we've now got the transit time in these same studies, average transit time. And it varies from, on the brown, quite rapid 43 to on the cabbage 64. And we can plot those on the rumen studies, and they very much are on a similar sort of line. So again, the human hindgut is behaving in a very similar way to other fermentation systems. And they're all towards the right-hand side of the turnover time because the transit times are sort of 40, 50, 60 hours, which is what you get in humans. That's the equation for the line. Okay, so in terms of biomass, the things that are important are substrate type. Well, I haven't really gone into that. And transit time, very important. Species mix has not yet been confidently shown. And I'm going to leave that to the microbiome people to show that. But I'm not sure it matters that much. Okay, right. How are we doing? This is the last bit. And this is just raises a question, and I'm not sure how we're going to answer it, okay? Right, okay, back to the biomass data. Here we are, our three diets and the amounts of biomass on the control and with fiber diets. And we know that the amount of increase is directly what we'd expect from the turnover time and the amount of fiber metabolized in these people. But all these subjects had a basal excretion of biomass. And this, the yellow columns show on the control diet, they've all got basal fiber biomass excretion. And that excretion is on average 14.8 grams a day. And we can use our equation to show that if you have 14.8 grams of biomass excreted each day and you've got a yield of 29%, you need 51 grams of substrate in your large bowel to produce that. on an average transit time. Fiber intake on these subjects on the basal diet was 16 grams a day. And that's sort of pretty average, you know, for most of us, I guess. No, not this audience. You're all big fiber eaters, but most people have, on a controlled diet would have this. So we've got a conundrum here. We've got lots of biomass coming out, and we've only got from the non-starch polysaccharide component of fiber a proportion of that. So what else may be important? Now, I've talked about surface effects, and I won't go into that anymore, and we won't go into transit anymore. But what other substrates could possibly be available to feed the biomass? And this is the classification of dietary carbohydrates, which WHO adopted, courtesy of Jim and I quite a few years ago now. And we've got to find carbohydrate. Other substrates are not so useful. And so here we have the sugars, the oligosaccharides, and the polysaccharides. And we've already talked about the non-starch polysaccharides. So the next thing we started to look at was starch, which at this time was thought to be rapidly and completely digested in the human gut. And of course, you now know, I think Lisa mentioned resistant starch, and it's a big story. The way we have investigated this was using this ileostomy model. This is a picture of the human gut with the small bowel, the large bowel, and the ileostomy person has no large bowel. They've had it removed because of ulcerative colitis. And they form a very good model for studying digestion. Right, so we feed different amounts of food or carbohydrates in at the top end and collect it from the end of the small bowel. And we started with the non-starch polysaccharides in oats, cornflakes, and white bread. And the amount fed and the amount recovered is exactly the same. All the fiber in those foods, and in fact in everything we ever looked at, goes straight into the large bowel. And that's what you would expect. And if we took some starches, like the starch in oats or cornflakes or white bread, that's all digested in the small bowel as well. So that's what we were taught. I mean, that all disappears. So there's nothing there, is there? Here's my favorite slide of all time. But bananas changed our minds. This is a slide of the ripening of bananas. And those of you who grow bananas will know that initially on the tree they're green, and then they ripen and become sort of yellow with black spots. And on the left-hand side, they're quite not very sweet, and on the right-hand side, they're very sweet. And that is because in bananas, the carbohydrate is present as starch. That's the bottom line. And as the banana ripens, the starch turns into sucrose. So your sugar in the green banana is 1% of the weight of the banana, but by the time it's ripe, it's 18%. This is the banana without the peel. So during banana ripening, the starch turns to sugar. And we fed bananas of different ripeness to 6 ileostomy subjects. And the yellow is the amount of starch in the banana when we fed it. And in some of the bananas that were ripe, it was only 1 or 2 grams, and in others, it was over 20 grams. So you can get a lot of starch, and no need for me to say that this is going to be called resistant starch in a single banana. And virtually, I think it's 80% of the starch, any starch in a banana, goes straight into the large bowel. Now, why? Okay, starch comes in many forms and in many different granule structures. Just there they are. And within the granules, starch takes on different crystalline forms. It's in a very elaborate and beautiful structure. And the X-ray diffraction patterns, last time I read about this, divided into A, B, and C types of crystal structure. The A are most cereals, and sweet potato and tapioca and roux. When I came down here 10 years after he'd done his sabbatical, he said you should look at taro as well. And Lisa tells me that's a Polynesian food rather than a New Zealand food, but a very starchy food. The B type of crystal structure is potato and banana. It's easy to remember, B for banana. And C are mixtures. Right. I can't go back. The A type of starch is well digested in the gut. Those are the cereals. The B types are resistant. The banana starch is completely resistant to pancreatic enzymes. Potato starch is, but we don't eat raw potatoes, we cook them. And that turns the resistant starch into a gelatinized, digestible form. But if you let your gelatinized starch cool, and this goes for either A or B, it always retrogrades into the B type. And then it becomes resistant. And after a lot of work, we developed, Hans Englist and I developed this classification of starches depends on their nutritional quality. And there is digestible starch, rapidly or slowly digestible. That has sort of implications for this glycemic index you all love. And then there's a resistant starch. And there are 4 forms. There's a physically inaccessible in the whole grains that Lisa was talking about. There's the B crystal starch, which is totally resistant. Starch which retrogrades. Any starchy food you allow to cool or put in the fridge will form resistant starch. Not all of it, but some of it. And there are modified starches. Okay, I'm just about finished. So we fed resistant starch to our ileostomy subjects from wheat, potato, maize-resistant starch, banana. It's all recovered in the ileostomy. Goes straight into the large bowel. And we also fed in our metabolic suite healthy volunteers diets containing significant amounts, 25 to 30 grams of resistant starch per day, and looked at how much came out in feces. If we look at the potato and banana on the top, all goes into the large bowel, but in virtually every subject apart from one, it's completely fermented in the large bowel. And this one is for the maize and wheat resistant starches. Not everybody will digest resistant starch in the colon, but most people will. So now those of you who know me and know how I hate systematic reviews will be surprised to learn that 12 years ago I actually did one. We now have the situation whereby we have resistant starches and they're fermented in the large bowel in a similar way to the non-starch polysaccharides. And if you want to call them all fiber, that's your problem. But they are separate. And these, at the time I did this, we did this for WHO actually, for that thing in 12 years ago. I found all the resistant starch studies that I could find. And I did this systematic review. We found a computer program you could download and just shove the stuff in. Didn't really understand it. But my feeling is that this shows quite clearly that resistant starch does affect bowel habit. And there's a sort of triangle there that tells you that. There are parallels here with the non-starch polysaccharides. And we know that it's also feeding the microbiome. So resistant starch is a substrate to add to our chart. One last thing, any other carbohydrates that might reach the colon, we've got resistant starch and NSP. They're what McCann's called the non-glycemic carbohydrates. The other ones are the oligosaccharides, and Michael talked about fructooligosaccharides in inulin. That's this group. They get to the large bowel, and these are studies using either ileostomies or long tube studies. And you'll see that these are the inulin. Oh, sorry, I've gone back here. I'm not sure I can change that, can I? Never mind. Can you go backwards? The bottom one. Oh, it does. Right. Well, just only one more slide after this. Yes. Right. But the message from this is that these 3 particular sources of oligosaccharides, you give them in the diet and you recover 88% of them. So these oligosaccharides escape. These are fructooligosaccharides, but the same goes for galacto-oligosaccharides. These do actually escape into the small bowel. But if you try and recover them in feces, the right-hand column shows you what the recovery in feces is. They never appear in feces at all. Not a single molecule ever appears. So you've got 100% fermentation of these oligosaccharides in the large bowel. But they don't affect bowel habit. That's a question to leave you with. And this is the other one of these systematic reviews I did. And I don't know what Lisa or Andrew would think about that, but the little triangle is quite close to this sort of no effect, isn't it? Yeah. Interesting. Now, why should that be? Very interesting question. Okay, I'm finished. This is the team in 1993. Myself, 2 microbiologists, Sheila Bingham, the nutritionist, and Hans Englis, the chemist. And Rue, tribute to him for the amazing work he did. And the last time I met him was in Athens in 1992, before, of course, he tragically died. So thank you very much for listening. There are a few questions left there, but this is an interesting part of digestion which we cannot ignore.